Today's talk, though, is going to be Dr. Jim Birchler, and he's going to be ably assisted by Dr. Ed Coe and Dr. Gerry Neuffer. So Jim is from a very small town in Illinois called Red Bud, Illinois, that I only know of because I had a student who was from there. His undergraduate was at Eastern Illinois University, and then he got his PhD at Indiana in genetics. After that, he did a couple of postdocs, one at Oak Ridge National Lab and one at UC Berkeley. He started his academic career at a small northeastern liberal arts college in Cambridge, Massachusetts, and then was recruited away from Harvard to come here, where he has been for the rest of his long and illustrious career. So with no further ado, let's please welcome Jim.
Thank you, Dee. The inspiration for this talk is in conjunction with the dedication of a commemorative bench to Louis J. Stadler, who was probably responsible for the great reputation that the University of Missouri has had in the field of genetics and also in plant biology in general. He started from his beginnings in Missouri that I'll tell you about. But we have on campus many spaces and buildings named after administrators and politicians, but we did not have any recognition of Louis Stadler and his contributions to the university. And so a few years ago, Ed Coe, one of my co-presenters, initiated the idea of getting a commemorative bench, which he will tell you about after my words. And so we will dedicate that after this presentation, and that was the inspiration to tell you a little bit about Stadler and what his contributions were to genetics and to the university as well. Many of the pictures that I have came from Ed Coe and Perry Gustafson, and many of the stories from Gerry Neuffer, who is also here. Gerry Neuffer was one of Stadler's graduate students. I've also relied heavily on Lee Cass, who is a science historian at Cornell University, who has been here on several occasions. Her specific interest is in Barbara McClintock, and Stadler and McClintock had a relationship that I'll tell you about that led to some important contributions to the field of genetics, and then also some work of Edward Calabrese at the University of Massachusetts in Amherst. So this is L.J. Stadler. He was a native of Missouri. He was born in St. Louis, Missouri. In his high school work, one would not have predicted that he would have gone on to much distinction, because his rank was 57 out of 69 students in his high school work. He got into, his father was a banker, but he got interested in issues of agriculture and came to the University of Missouri to study agriculture, but decided he wanted to have fun in the sun, I guess, and so he transferred to the University of Florida and got his bachelor's degree from Florida. But as fate would have it, his person with whom he was working at Florida accepted a position here at the University of Missouri, and so he came back to Missouri. As you will see, he very often did. He tried to escape many times, but he always came back. And got a master's degree in field crops, and then he tried to escape again by enrolling in graduate school at Cornell University with distinguished maize geneticists there. But his talents were not appreciated at that time, and he was sent packing. So he came back again to Missouri and received his PhD in 1922. He joined the faculty. We here at Missouri always appreciated his talents. And he published on various agronomic problems for the first few years. And then in 1925 and 26, he got a fellowship to go to work at Harvard with E.M. East and at Cornell with R.A. Emerson, who were both prominent plant geneticists at the time. And it was, in fact, Emerson who had sent him packing a few years earlier as a graduate student. This probably was a very interesting coincidence that he was there at those places at that time, which we'll come back to in a minute. So his first major contribution was that X-rays would have a mutagenic effect. The first recorded data that we are aware of that he used X-rays was on July 30th, 1924. He and Hermann Muller, who was a Drosophila geneticist at the University of Texas, had used X-rays. They were trying to increase the rate of recombination. This is still an issue in crop breeding because that slows down the way that you can incorporate new genes into particular varieties. And so in that time frame, they were both working on this. Hermann Muller published a paper in Science in 1927 revealing this discovery, and Stadler, in just a few months, dated in 1928, also in Science, published that X-rays would cause mutations in barley as well. So what to sort of set the context, before this time, geneticists had to rely on spontaneous mutations or natural variations to do any kind of studies and to understand anything about the processes of genetics. Of course, Mendel and Morgan and various other practitioners of the early days of genetics used those spontaneous or natural variants to great effect. But this discovery of Muller and Stadler to experimentally get alterations in the genetic material was a very significant advance that basically exploded the field, and it was recognized at the time of this. So this is a picture that Ed Coe supplied, and Ed has a nice quote about this. Here's Stadler at his X-ray machine. Ed says, "This machine, when I first saw it and used it in 1955, was a marvel of levers, gauges, and barely controlled sparks and ozone." So to give you yet another indication of the impact of this finding is that Hermann Muller won the Nobel Prize for the discovery that X-rays cause mutations. And many people have thought, and even just in the last year, I read a paper suggesting that perhaps Stadler should have shared in that Nobel Prize. Interestingly enough, Muller and Stadler had sort of a rivalry going. Muller said that X-rays were causing genes to be changed, and Stadler said over the course of his work that genes were being deleted and there were alterations in the genetic material, and it wasn't really changing the gene. Now, in the molecular era, many of the mutations that Stadler recovered, we now know are in fact deletions and deficiencies of various parts of the chromosome. And so we now understand that in fact, Stadler had the best interpretation of what was going on beyond what Hermann Muller had postulated. So part of the reason that Stadler came to this conclusion was that he had also studied spontaneous mutations, and he realized that what was coming out of the X-ray experiments were very different than what would pop up spontaneously. And he tested some very, very large populations of maize. Some of his data actually have a million corn kernels. I can't imagine this. I work on corn myself. I can't imagine looking at a million corn kernels, but he has some of the largest data sets on spontaneous mutation rates that are still quoted today. And he developed what is called the specific locus test of taking many different gene mutations and then looking for the rates of mutation against that particular set of mutations. This is still used today in studying carcinogenic activity of various chemical compounds, primarily in mice, but the principle of it has been applied for that purpose. Stadler also studied, eventually evolved into studying ultraviolet light and its mutagenic effect is in a contrast to the X-rays. And there had been some reports previously that X-ray would kill bacteria. And so Stadler then took it upon himself, together with a physicist in the physics department here, Uber, to examine what wavelength of ultraviolet light would be, in fact, effective at causing mutations. And he found that 260 nanometers was the most effective mutagenic wavelength. And he actually predicted that DNA was the genetic material, and that, of course, UV was different than X-rays in what kinds of mutations were found. So this was published in 1942, and previous to this, people thought that protein might be the genetic material. And biochemists didn't come to the conclusion that DNA was the genetic material until 1942. So Stadler and Uber actually, from these UV irradiation experiments, correctly predicted that DNA would be the genetic material. So that was another interesting foresight that Stadler had to the field of genetics. This is a picture of Stadler in about 1940 with Herschel Roman, who was one of his graduate students. Herschel Roman then got his PhD from here at Missouri. He went on to do much of the work, initial work in yeast genetics. And yeast now has proliferated to be a very important genetic model organism. And it was to no small effort that Herschel Roman was involved in those early efforts to use yeast as a genetic model. Now, as I alluded to, Stadler had a big influence on Barbara McClintock, who he recruited here actually on 2 occasions. And in some respects, one might say that Stadler was sort of the midwife to McClintock's discoveries of transposable elements, which eventually led to her winning the Nobel Prize for that discovery. So McClintock had gotten her PhD at Cornell, and this is Emerson that sent Stadler packing, but then took him on as a research fellow later. This was a distinguished group. Also in this group was George Beadle and Marcus Rhodes and Charles Burnham. And here's McClintock. What McClintock had worked out as a graduate student and then as a research fellow at Cornell after her PhD was a way to identify all of the 10 chromosomes of maize and was able to associate some of those with the genetics that was known. And so this was an important contribution that in working with the other people at Cornell, she made a number of very significant contributions to genetics. It was at this time that Stadler had visited Cornell, and he had become acquainted with McClintock at that time, and that led to their association. So McClintock had her PhD in 1927, and she made these studies on the chromosomes and their identity that was known, and she was recognized for showing that recombination, or the exchange of pieces of chromosomes, genetically and on the chromosomal level were identical. And so this was recognized in 1931 as a major contribution to genetics. It's in practically every genetics textbook to this day. So Stadler invited her, since she was good at looking at chromosomes, he was interested in having her examine this X-irradiated material. And we have this letter, this is from Ed, that I think came from the archives at MU, yes, came from the archives. And you can see it was dated in 1927, and this is a response to L.J. Stadler from McClintock. Basically, many thanks for the idea that we should look at the chromosomes in this X-irradiated material. So then in 1931, this is a letter from McClintock to Charles Burnham, who kept all of the letters that McClintock had ever written to him, and they are available at the archives at NIH. And she notes that in writing to Burnham that Stadler had arranged for a fellowship for her to work at Missouri for $500 for 2 months in the summer of 1931. So she said, I can work on whatever I please, but he wants me to tackle some deficiency stuff. I rather think I shall go. So she came here. She wrote lots of letters to Burnham about her experience at Missouri. I'm not going to go into those. They're kind of amusing in many ways, her impressions of the University of Missouri. But she came here in 1931. And Stadler had her analyze on the chromosomal level many of the materials that he had been X-irradiating. From this material, she described chromosomal changes, pieces of chromosomes that had been deleted, pieces of chromosomes that were exchanged, chromosomes that now had formed into a circle or a ring chromosome, pieces of chromosomes that had been switched around in their orientation, and those that had formed bridges during the process of cell division. So this was published in a very long but not very visible publication in the Missouri Agricultural Research Station Bulletin in 1931. But it set the stage for many things that McClintock would later study and analyze. What Stadler had set her out to work on, that deficiency stuff that she referred to, was to understand what Stadler had published upon the year before called Deficiency Recovery, where he had X-irradiated corn that had a dominant marker, and then one could see areas where the dominant marker was missing, but in part of the plant or the part of the kernel, there was a recovery of multiple genes that are next to each other. And so this was what she was supposed to work on. But years later, she refers to this, and she was never able to solve what was going on. That's okay, because no one to this day understands what's going on there. And so I like to joke that if your postdoc advisor gives you an intractable problem, there's still hope for a great career. Advisors all think that's funny, postdocs don't. So as I mentioned, Stadler and McClintock had this relationship, and Stadler had recruited her again. So McClintock went off to Caltech to do some more work. She went to Germany for a while, and Germany was at the time when Hitler was coming to power. She did not like the political atmosphere there. The people that she went to work with had already come to the United States. So she was there for a short while. She went back to Cornell, but then Stadler arranged a search for a faculty member and recruited McClintock again to Missouri in 1936. With the intention to do more work on this X-irradiated material. Then she moved into the Department of Botany when this appointment was renewed. And as an interesting note, in 1936, Stadler also tried to recruit George Beadle to the University of Missouri. He had an offer also from Harvard, and so he took the job there. And I would note that he also won the Nobel Prize for the one gene, one enzyme concept. So Stadler had great taste in those individuals that he was recruiting. He also recruited Ernie Sears in 1936, and I'll tell you more about him later. So when McClintock was a faculty member here, the thing that she discovered is what she called the breakage fusion bridge cycle, or BFB cycle, which one can visualize here by these mosaic corn kernels. And the process is that chromosomes will break and join and break and join and break and join over and over again under certain circumstances. So what led McClintock to realize from those studies was that there's a special property to the ends of chromosomes, which is called the telomere. So the telomere eventually was identified as a special structure. You may be aware of it in studies of aging and the length of telomeres and so forth and so on. But it was McClintock who originated the idea that the ends of natural chromosomes had some special property to them, which we now know and we call the telomere. This is material that has from my lab when Fangpu Han, who is in the audience here visiting from his position in China, material that McClintock developed while she was at the University of Missouri to make this bridge type chromosome that will initiate this cycle of breakage and fusion and breakage and fusion. So we still have some of McClintock's materials that she constructed while she was here. So this is a portrait of the genetics department in 1938 on the steps of Lefevre Hall. Here is Stadler, and here is McClintock. Here is Ernie Sears. There is Herschel Roman that we've alluded to also. So this was quite a distinguished group that was present here in that era. So here is a picture from the field, the cornfield. This is Stadler, and here's Ernie Sears, and here's Roman. This is John Launen. He was a graduate student with Stadler, and I mention him because he was Ed Coe's PhD advisor at the University of Illinois. So both Ed and Gerry have a connection with Stadler. Gerry being a graduate student and Ed being a grad student academically. So we have a picture here of two students that were with McClintock while she was here at the University of Missouri. Ed Weaver, who got his PhD working on corn, and Helen Krauss, who worked on a fly, Sierra, looking at chromosomes that were rearranged. So Helen Krauss made a contribution to this book called The Dynamic Genome about McClintock, since she was a graduate student of McClintock while she was here at Missouri. And I just want to read a couple of paragraphs, one about McClintock and one about McClintock and Stadler. So Krauss writes, McClintock's one diversion that I was aware of was having lunch downtown daily at Jack's Latch Cafeteria, followed by a visit to the local weather station located on the second floor of the post office building. This building is on Cherry Street. It was the federal building, if you're familiar with Columbia, and then it was part of the U Museum, and then it was part of Columbia College. So that was where she went daily to talk to the weather station people. She was interested in weather forecasting and tried to convince the two men who worked at the station of the superiority of forecasting by air mass analysis. I accompanied her to the station on several occasions and thought the men looked forward to her daily visits. They were cordial and shared with her their latest charts and data. Occasionally, she joined a group of us for supper at the Virginia Cafe, where you could get an excellent Western steak for 50 cents. The Virginia Cafe, I figured out where it is. So if you know where the Top 10 Wines is on Ninth Street, that's about where the Virginia Cafe was at that time. So then Krauss writes about the finishing up of her PhD with McClintock and her interaction with Stadler. I'll tell you about it in a minute. McClintock, of course, left the University of Missouri, and Krauss writes, most of my research for my thesis was completed by Christmas 1940, but all of the data had to be summarized, salivary chromosome maps drawn, photographs made of each translocation, and the writing of the entire text accomplished. I began to get leverage on some of these tasks during the spring of 1941 and received a boost when I discovered that McClintock had given me an A in cytogenetics. I was settling in for the hard pull when McClintock came by and said she was leaving the University of Missouri for good. Her car was packed up and she was going back east. I was so upset by this startling announcement that I could hardly comprehend the situation. Suddenly she was gone, never to return. Although she did, I'll tell you later. During the ensuing 5 months, I worked doggedly on the thesis. Stadler was helpful in every way possible. When I learned that the American Association of University Women had awarded me a fellowship, he arranged a postdoctoral appointment with Theodosia Stubzansky at Columbia University, who was a very well-known Drosophila geneticist. Getting the thesis approved by Miss Guthrie, who you saw the picture of her, was a colleague of McClintock, was tedious and exhausting, to say the least. Finally, after more than a month wasted in trivial changes in punctuation, Stadler told me to tell her to take it or leave it, and I was going to New York. He assured me that I had his full support in this confrontation. He also advised me, jokingly, to remove my lipstick before the encounter and to appear as bedraggled as possible. Anyway, she passed her exam and went on to great things later. So in 1940, Stadler was not necessarily trying to escape from Missouri again, but was trying to be recruited by California Institute of Technology, Caltech in California. And he was on a visit there, and they offered him a faculty position. And the MU administration suggested that they would just get rid of the whole genetics group if Stadler took that position. Of course, McClintock was one of them. So here you have, in 1940, McClintock is writing to Stadler at Caltech, and she has a very chatty letter writing to him. Everything seems wonderful. They're building Curtis Hall, if you know where Curtis Hall is, and everything seemed to be going good. And so, interestingly enough, in the year, almost to the day, February 10th, 1941, this is a letter from McClintock to Alfred Blakesley at Cold Spring Harbor Laboratories outside of New York. Blakesley had invited her to spend the summer there doing some chromosome work. And so she took a leave of absence from her position at Missouri and accepted a visiting researcher position at Columbia University, where Marcus Rhodes, part of the group at Cornell, and her good friend was at Columbia University. And so she was intending to return if she could not find another position. Here she is. This is a picture that I got from Cold Spring Harbor Laboratory shortly after she arrived there from the University of Missouri. So once this had all transpired, Stadler wrote to Rhodes at Columbia and told him that McClintock would receive tenure, but Dimmerich became the head of Cold Spring Harbor Laboratories and offered McClintock a visiting investigator position, and then he offered her eventually a permanent position. University of Missouri countered with an offer of a raise and promotion to tenure. Her salary then would have been $3,990 per year. In something that I just read fairly recently, people at Cold Spring Harbor Labs was organizing some information about McClintock, and they wrote to me about her experience at the University of Missouri, and in some of that information, the offer at Cold Spring Harbor was about the same amount of money, and her potential colleagues at Cold Spring Harbor were outraged at the high salary she was being offered. So it is sometimes noted in some of the biographical material about McClintock that she was denied tenure at MU. This is in fact absolutely incorrect. She was offered tenure and in fact a raise to try to convince her to come back to the position. Apparently she weighed this very carefully but decided that the freedom that she had just to do her own research at Cold Spring Harbor was more suited to her personality. Shortly after she went to Cold Spring Harbor, she discovered transposable elements, pieces of DNA that jump around the genome, makes up most of our genomes actually, and this was from material undergoing this BFB cycle. And this discovery went on to lead to her being awarded the Nobel Prize in Physiology or Medicine in 1983. This is a picture of McClintock's corn kernels from Cold Spring Harbor very early on in her studies of transposable elements. The little spots here are instances of where a transposable element has jumped out of a gene or inactivated it. And that is the kind of data that she used in deciphering transposable elements. This is McClintock here on the day that she was awarded that Nobel Prize, and in her Nobel Prize speech in Stockholm later that year, she starts it out saying that her journey to the discovery started at the University of Missouri in 1931 when Stadler had invited her to look at material that had been undergoing X-ray mutagenesis. And so that obscure publication about all of the different kinds of rearrangements that X-rays would produce was the initial seed that laid the path to her discovery of transposable elements. And then she also mentions her time as a faculty member here as being instrumental, indeed, because that was the material in which that she developed here at Missouri was where she found transposable elements eventually. After Stadler had passed away, I'll come back to this later, there was a Stadler symposium set up in his honor, and for one of those, McClintock came back, and this is a picture from Ed Coe. This is Gerry Neuffer in 1978, Ed Coe's graduate student Sheila McCormick, and here is McClintock. This is where McClintock's laboratory was most of the time when she was here. She spent a short time in Lefevre Hall, spent most of her time here in Stewart Hall, and then her last year in Curtis Hall. It's reputed that she forgot her keys one weekend to the building, and she shimmied out on this ledge, probably balancing herself on the lamppost, and let herself into the window of her laboratory. So as we mentioned, another individual that Stadler recruited to Missouri was Ernie Sears, also in 1936. And Ernie Sears worked on wheat genetics. This is a slide that I got from Ed Coe detailing many of the contributions that Sears made to wheat genetics. Before Sears came along, it was very difficult to do any genetics in wheat because wheat is a hexaploid. It has 6 copies of every chromosome instead of the 2 that most diploid organisms would have. And so he worked out a number of ways of manipulating this situation so that genetics could be conducted in wheat. And these stocks still exist and are used worldwide in studying the genetics of wheat. Of interest here is that he showed how one could transfer disease resistance genes from a wild relative into wheat by this chromosome engineering. And this kind of technology is still used today for that purpose. He was awarded the Wolf Prize in Agriculture in 1986 for his development of all of these materials that led to being able to study genetics in wheat. This is Ernie and his wife Lottie, who was also a distinguished geneticist, worked on much of the wheat work and also studied the chromosomes of Arabidopsis, was the first person to do that work as well. And here's a later picture of Lottie and Ernie Sears in their laboratory. So then I'm going to move to my co-conspirators in this talk, Gerry Neuffer and Ed Coe. They filled Stadler's position and are still with us. Gerry is 96 years old. He confirmed to me, and he will give a few words here in a few minutes. And Ed is 91. They still are active and interested in what they are working on. So Gerry was a student of Stadler, a graduate student, and Ed Coe, as I mentioned, was a grad student of Stadler, being a student of John Lonan in Illinois, who was a Stadler graduate student. So here's a picture of the genetics department 50 years ago. Here is Ed, and here's Gerry, here's Ernie Sears on the steps, I believe, of Curtis Hall at this point. Yes, that's correct. So they are there. So I want to just mention some of the contributions that Ed Coe has made. He discovered paramutation at the B locus in maize. He studied the biochemistry of pigment production. He found a stock that makes haploids in maize. He was instrumental in organelle genetics and worked with Kathy Newton, who is here and is on our faculty as well. He was involved in studying developmental mechanisms, molecular mapping tools, early maize genomics, and database development. And just to give you an idea of a collection of some of the things, this is paramutation. These are pictures I got from Ed a long time ago. This is heavily pigmented cobs of ears. And what he found is that he found a case where paramutation violates the rules of Mendel, so that if you have a particular version of a gene that is silenced, it will transfer that silencing to its partner in the cell, and its partner then can pass it on to generations after generations. And so you may be familiar with epigenetics. It's a very popular field these days. Well, Ed was one of the initiators of the whole field of epigenetics. These are ears, a bountiful crop from haploid maize plants. You don't get much kernels from a haploid maize plant, but if you double the chromosomes, you have a line of maize that is totally homozygous for every gene. The doubled haploid breeding, all of the stocks that are currently used are derivatives of Ed Coe's, what he called stock 6. It was his 6th stock that he ever had in his collections. In my travels with talking to people at Monsanto and Pioneer and Syngenta, they've all changed their names, but those are the old names of them. They literally make millions of haploids every year in developing new lines of maize, and all of them use Dr. Coe's, or derivative of Dr. Coe's original stock 6. Just in the past year or two, the major gene involved in this has been identified molecularly, And so people are interested in expanding the ability then of making haploids with that molecular information from Dr. Koh's original discovery. Gerry Neuffer studied transposable elements, and from that he got inspired to develop chemical mutagenesis in maize and characterized literally thousands of mutations, and was very generous in passing them out to colleagues here at the University of Missouri and beyond. Some people who were at Missouri who studied them was Don Miles, who's retired, Bill Sheridan, who has moved on. Current faculty member Paula McSteen has used some of his mutants. The disease lesion mimics were of intrigue to Gerry, who had studied them, and then Tony Kasich, who is right here, is studying them now, and one of our current colleagues, David Braun, is studying some sugar transport mutants, and there are many, many others that have benefited from Gerry's contributions. This is just a picture of one of the transposable element systems that Gerry worked on, the so-called dotted system. And this is an example of a disease lesion mimic mutant. These are dominant, and this was a great intrigue to Dr. Neuffer, and he studied many of these, as have other individuals. Ed and Gerry were organizers of a compendium of the genetics of maize, the mutants of maize, together with Susan Wessler. This, of course, was the initial compendium of the genetic changes that occur in maize, and this has been expanded from this initial organization. So here's another picture of Ed and Gerry. They arranged to nominate Barbara McClintock for an honorary doctorate from the University of Missouri, which was awarded in 1968. McClintock received honorary doctorates from many institutions over the years, but the University of Missouri, at the instigation of Ed and Gerry, was one of the first to give her an honorary doctorate. So one of the other individuals that I want to note is George Redeye. He came to MU in 1957 from Hungary. He was there was political turmoil at the time, and so he came to the U.S. It was after Stadler had passed away, but I'm sure he was attracted to the genetics group that was here, and he brought with him seeds of Arabidopsis. And for many years, 20 years in fact, he was the sole person working on Arabidopsis in the United States. Today, there are hundreds, and that is probably an underestimation. There's probably thousands of people who work on Arabidopsis today. And the inspiration for that was in 1975, he wrote a review about using Arabidopsis as a genetic tool. And this inspired many people at the time to pick up this this plant that has a very short generation time and is small, and so you can grow many of them inexpensively in a rapid time. And that inspired and laid the foundation for an explosion in plant biology. The Columbia strain, those of you who are familiar with Arabidopsis have heard of the Columbia strain. That's named after Columbia, Missouri, and it came from George Redeye's collections. Here is George Redeye with Barbara McClintock when she visited in 1978 in Ed Koh's lab, and Ed has noted here that genetics is far out. I also want to mention Perry Gustafson. Perry is with us here today. Somewhere I saw him earlier. Perry arrived in 1981, and he made many important contributions to the introduction of genetic material from wild species, and also for understanding aluminum tolerance in rye. He was the organizer of the Stadler Symposium for many years after George Redeye relinquished that role. And he was also instrumental in the organizing committee of the plant and animal genome. He's been a strong advocate of world food security and travels all over the world in that capacity. So I want to return now in the final minutes to Stadler. As we heard from the comments of Helen Krauss that he was an inspiring leader to graduate students, very supportive of graduate students. In his obituary, Marcus Rhodes wrote that he had an inner serenity which gave him a poise and suave dignity that was never ruffled by an unexpected or disappointing turn of events. He was interested in humanitarian issues and the role of science in society, and he often testified before Congress on various scientific issues. In his interest in the societal issues, he joined a number of groups that were trying to be of assistance to scientists who were under siege. And one of those was he signed on and contributed money to relocate academics from concentration camps in France, people who were fleeing Franco's regime in Spain in the 1940s. He also joined organizations to help relocate refugees from the Nazis in Germany to the United States and contributed money to that. His wife, so these organizations had criticized some scientists, and they said, oh, those organizations are just fronts for the Communist Party. And as it just so happened, Stadler's wife had subscribed to the Daily Worker for a year, just as of interest to her. And what this resulted in was that in 1948, Stadler applied for a passport to go to a meeting in Europe, and he was denied a passport because he was suspected of being a communist sympathizer, and he was subjected to a loyalty investigation, and it took about a year and a half, and his position in the USDA was potentially under threat. He might have lost his job, but eventually, he was cleared after an extended investigation for loyalty. This is a picture of Lou Stadler and 3 of his 6 children. David Stadler went on to be a distinguished geneticist in his own right. So here is Stadler irradiating some corn plants. This is Stadler. He had this machine here, and here is the critical part of this machine. And Mel Green, a professor here at MU, he was initially an instructor and then became an assistant professor from 1945 to 1950. Mel Green worked on Drosophila. At the fly meetings where I was going, he would always say, Birchler, come over here. I want to tell you about Stadler. And so he was a great admirer of Stadler. And he would tell me a particular story about this machine. He would always say, I don't think I've told you this story before, and I think I've heard it 5 or 6 times. So this is the important part of it. And so Green was working with Stadler, and they were going down the corn row, irradiating corn. And when it came time to harvest, they didn't see any kind of changes. They said, well, what's going on there? Because this was now in the era of 1945 to 1950. And so the initial discovery was in 1928, publication of it. So what was going on? So they checked all of the cords and all of the levers and the buttons and everything looked like it was working fine. And then they looked at this part of the machine, and it was pointing upward. And as they were going down the row, they were irradiating the sky. So those errant X-rays probably led to Stadler being diagnosed with leukemia. Leukemia is one of the consequences of exposure to X-rays and irradiation in general. Marie and Pierre Curie both had leukemia, and their family members that worked with them, all of them had leukemia. So with it's always difficult to assign a cause in a single case, but it seems reasonable that probably Stadler's leukemia was due to his exposure to X-rays during his studies. He was in remission for many years, but in 1954, at the age of 57, he passed away. The University of Missouri tried to recruit McClintock to take his place. She declined and stayed at Cold Spring Harbor. John Lonan, who was Stadler's student, accepted the position for 1 year, then went back to Illinois, and then Ed and Gerry filled that position, and they are still here with us today. So those errant X-rays then, Stadler contribution to the University of Missouri, attracting very excellent individuals, training many excellent individuals, certainly led to the initiation of a great tradition in genetics and in plant biology here at the University of Missouri. So with that, we can conclude our discussion of L.J. Stadler. Thank you very much.
Alrighty, if you have any questions, please raise your hand and I'll bring you a microphone.
What I want to take this up, I had an ulterior motive in volunteering for to give this talk in addition to the Stadler bench, and that is that I wanted to on campus note that Ed Coe and Gerry Neuffer are the 2018 and 2019 Emerson Lifetime Contribution to Maize Genetics awardees, and they will receive their award next March in St. Louis at the annual Maize Genetics Conference. So Gerry was a student of Stadler, and so if Gerry would like to say a few words about Stadler, we would appreciate that. Gerry, you want to?
I don't use.
Yeah, just hold it close. Testing, yeah, it's fine.
Okay.
Hold it close. Close to your mouth, Gerry. It's good.
Okay. Can you hear me? I can't. Now can you hear me?
Yes.
We have a trouble in our house. 3 people there I can't hear, and we don't communicate very well. I hope I can communicate with you. I laid awake a bit last night thinking what I was going to say, and it's true. He's told all there is to know about Stadler's work, I think, and about mine and about Ed's, and we don't have much to add, but I want to tell you something. I'm a grateful person because Stadler was responsible for almost everything good that's happened to me in years. I was born the year that he came to Missouri, and I was invited. I had an opportunity to come out here to go study either the Ames out to California or a couple other places back to Cornell, but I decided to come here because they offered me some money as an inducement to come with no place to live, and it was pretty hairy at first. And the Stadlers made this thing easy. So I'm going to talk just a minute or two about my relationship with him. Sounds like I'm talking about me, but it's really I'd like you to see what happened. I came here as a student who never studied in high school because I had to ride all back and forth on the bus every day, and couldn't take books home to study because I had milk cows at night and morning. So I was not a student really. I went away to the University of Idaho, and I took genetics and got a B in the course. I loved it, but I got a B because I didn't study. And I came here, and I was not very well prepared intellectually and educationally. I was not prepared to take this position. But Stadler took me on, and he did some things for me that I can always be grateful for. He took this green student in and made a special effort that I should succeed. Well, Stadler taught me how to give a seminar. He said, I give the first seminar, and he said, I said, how do you do that? He says, Gerry, just talk to the dumbest, don't talk to me, just talk to the dumbest student in the audience. And you make it all right. And so I got through there. And then another time he told me how to write a paper and did all these kinds of things. I couldn't understand why he kept me aboard, but I finally figured it out. Stadler was ill, and he had all this project going, and he had all these people that were important, and he needs somebody back home to look after the goods. And so I inherited the job. And Henry.
It's okay. Just keep it up there.
Okay. I and Henry Lee and Gus Dosage was the. Okay. All right. I got it better. I can see there's some glazed over people back there, but Henry Lee and Gus Dosage and a few other people that Stadler took aboard. We ran his project. Now Stadler liked to have things happen, and one time he said we were discussing how we figure out what was truth in the matter, and he says, Gerry, he says, if it works, it's true. Oh, Gerry, I'm going to get you a book. And so he picked up the receiver and called over to the bookstore and send this book over to Gerry Neuffer, Bridgman, the Logic of Modern Physics. He says, if it works, it's true. And so that's the way that Stadler did, and that's the way I did it. And we looked at things, and that was right at the time when he had decided that X-rays do not cause mutations, but they cause destruction. But he wasn't able to prove it quick enough, so he didn't get a Nobel Prize because it takes a long while in barley and corn to get a progeny, and alpha takes only 11 days. And Muller was able to prove the point, and he got the prize. Nowadays, they give it to both of them. So Stadler was this way about things. He liked to grow corn at all times of the year. And one time he says, Gerry, we got some corn out there between the greenhouses, and it's September and it's ready to pollinate. September 23rd, it's ready to pollinate. We're gonna have a frost tonight. Do you know what to do? I said, sure, I know what to do. I said, you just spray it with water. He says, how do you know that? Just have ice all over it. I said, that's what we did out in Idaho. We had tomatoes, and the only way you got a crop before September is to make sure you got it out before the frost. Well, one time we had some frost coming on, and my dad says, I remember in physics that when water freezes, it gives off heat. And let's just run a little water down the rows all night long. And sure enough, next morning, there's ice all over everything. But we had tomatoes, and we paid for the land that year. So I said, I just sprayed. Well, I came out, sprayed that corn, and next morning there was icicles hanging for all the tassels. But the ice melted away, and I made pollinations from that corn. And Stadler appreciated that, I think. And I remember he got ill, and we had a new farm out there, and we had to plant corn in 1954, and that was the year of July 1954. We had the first 14 days of July were above 110, and we had a cornfield growing out there, and phone. We had, we had, we had a cornfield growing out there, and and we had that corn, and he'd bought a little pump with a little outlet about that big and 3-inch pipe, and he said, Gerry, will you irrigate that corn? We had a little water in the pond, and you know we had the only corn in Boone County, and that's because Stadler asked me to do that, and I knew what to do, and that was a nice thing to do, and he appreciated it. He was always that way with me, and then the thing he got. I never did work for that. Wearing my finger out here. Okay, now you're all right. We have this. Well, I was going to story. Well, I tell you stories for hours about that he did. One of the things that Cornelia, his beloved wife Cornelia, was a wonderful person. She got involved in all kinds of good things. She thought communism was a good thing, but she was the thing that she saw that my kids didn't have shoes. I was a graduate student, had 4 children, and no car, and an income of $100 a month. And she saw this, and so she bought all the family shoes, including me. And she was always doing things like that for us. And my second son was a baby, and my wife was changing his diaper, and she went to the bathroom to something come back, and one of the safety pins are missing. And when she couldn't find it, and she realized he was kind of coughing, and so we knew he'd swallow the safety pin. So Stadler let me take the car and drive the car down to St. Louis, take my son down to Barnes Hospital to have that safety pin turned over and pulled out. And so these things happen. Well, when Stadler got to near the end, he needed somebody to take over. They invited John Lonnon to come here, and he left and went away. And finally, when he got bad, he was in the Barnes Hospital in St. Louis, and he asked the dean to come down and talk to him. And the dean went down there and talked to him, and I don't know what they talked about at the time, but I figured it out later. Stadler had the dean promise to take care of his project. And one of the things he wanted to do was to have me and Ed Coe. Ed Coe was to take the USDA money and be hired, and I'd take the university money and be hired, and so the two of us would replace Stadler, and we made a mighty fine combination because we were never threatened by each other. We were good friends and we worked together, and in fact, my work led to wanting to do what Stadler had wanted to do, to show how genes can be changed. Mutations can occur, artificially occur, as well as by spontaneous means. And we found out that there was a chemical called ethylmethane sulfonate, and Ed Coe said, well, we can put that chemical in paraffin oil and do that, and we can make lots of mutations. Well, when we did this, it's just like pointing a pistol at a mountain, and the mountain, pull the trigger, and the mountain explodes. I suddenly discovered I had more corn mutants in my laboratory than anywhere else in the world, because it increased the frequency of mutation 1,000 times, 1,000 times, mind you. Before you normally think of getting 2 or 3 during your career, 2 or 3 mutations you find spontaneously. I thought about 2 or 3 a week, and I had more GMOs in my laboratory than anywhere else in the world. So,
Thank you, Gerry.
I'm grateful. That wasn't what I prepared, but
So Ed is going to tell us about the Stadler bench at this point and tell us the proceedings that we will have for that.
It's a bottle. It's fine.
It's on?
Try saying something and we'll see if we can hear it.
Is this working? All right. I'll be just a few minutes. I'm impressed, of course, with what has happened here after Stadler, and Gerry just demonstrates it extremely well. Of course, I'm impressed with the way Jim covered what his talents and his experience and knowledge show. It was a very good presentation, Gerry.
I'm honored by that.
This guy over here. I do thank Jim for that. Thank you to everyone who came to participate in this event. It's a very special opportunity for me because for 63 years I've realized I walked into a treasurable situation. And it was because of what Stadler had set up. And Gerry had the role of keeping everything going. He had the resources available to him. And I should say thanks to everyone who donated to the bench that we will have the opportunity to go to in just a little bit. Dr. D, Dr. Cornelison, it's a pleasure to have this opportunity as well. And the development office here at the university and the Mizzou Garden have been very helpful. As for myself, I arrived here, as you know, in 1955, at the time Dr. Neuffer placed his foot in one of those big shoes. Stadler had two great big shoes, as you have had described. Gerry put his foot in one of those, and I put my foot in the other. And you've heard about trying to do something important with one hand behind your back, or tied, and that's how we went about our science. But we had to lock arms on a lot of things to do it well. It was has been quite a partnership. One more thing before I introduce the bench business. Stadler had a special talent as a scholar scientist. That's evident from what you've heard from Jim and Gerry this morning. I heard him only once give a series of talks at the University of Illinois when I was a grad student. He would present observations and he'd follow with suggested explanations, then with possible experiments by which explanations could be eliminated, not to prove something, but to get rid of the wrong explanation. And I thought I gained great insight about how to do science from that at that time. About the bench. The location, as in the picture that Jim showed, is right between Curtis Hall and Stewart Hall, out in front of that renovation there that's been going on for some time. It's finished. And it's along the walk that goes across in front of Stewart and Curtis. Curtis was a building initiated by Stadler's obtaining of a grant and support from the state, and so that's a monument to him. It doesn't have his name. What we'll do, if you're prepared to walk over to see the bench, it's covered right now with the Mizzou flag or banner, and we'll unveil it over there. The easiest way to get there is out the side exit, out this way onto the walk that goes on up past the Sears greenhouses and around Curtis Hall to in front of Stewart and Curtis. We won't take long. We'll just celebrate this moment right there at that time. I'll join you there as you travel. Thanks very much.
So anyone is welcome to come join in the festivities. I know I want to have a picture with Ed and Gerry sitting on the Stadler bench, and so I welcome anyone else who wants to join in that kind of festivity to join us as we head in that direction. So thank you very much for your attention today.